Welcome to the Deep Dive, where we plunge into the incredible, often unseen diversity of life on Earth. Today, we're zooming in on a microscopic world that, well, constantly challenges our common perceptions of what living things can do. We're focusing specifically on some truly extraordinary fungi. Our mission for this deep dive is to explore specialized fungal forms, the anamorphic fungi. Now, anamorphic simply refers to the asexual or imperfect stages of fungi. Often these are the most visible or impactful part of their life cycle. And these are the stages that have evolved these just mind-bending adaptations to thrive in very specific, often challenging environments.
Exactly. And we're drawing from some fantastic sources today, notably excerpts from Introduction to Fungi. Now, this book is packed with scientific detail, but our goal here is to unpack it for you, our listener, especially if you're, say, a college student grappling with these complex biological concepts. We really want to translate that dense scientific prose, make it clear, vivid, memorable, help you truly imagine this unseen microscopic world.
Absolutely. And we're going to tease apart two main fascinating areas today. First, we'll meet the fungi that hunt microscopic worms in the soil. Yes, they literally set traps for them. It's amazing. And then we'll dive into a group of fungi that have become, well, masters of the aquatic world. They've developed these stunningly unique spore shapes to survive in fast-flowing streams and stagnant pools. Okay, let's unpack this. All right, let's start with the micropredators of the soil, what scientists call nematophagous fungi. Now, when we talk about nematodes, these aren't just any worms. They are an incredibly varied group of invertebrates. You find them almost everywhere, right? Soil, water.
Pretty much everywhere, yeah.
Yeah.
And most free-living ones just feed on bacteria, nothing too dramatic. But some are serious parasites of animals or plants.
Right, like those economically important pests, the root knot diseases and root cyst diseases. They cause huge problems for crops.
Exactly. Their mouth parts are even modified into these sharp stylets to pierce plant tissues. And some, like the cyst nematodes, turn their whole bodies into these hardened, egg-filled cysts. Incredibly resilient things.
Wow, so a pretty serious threat, and fungi have stepped up.
They certainly have. In response, fungi have evolved this range of ingenious mechanisms to attack these nematodes. And what's truly remarkable here, as you dig into it, is that these attack strategies fall into three broad categories. And they often show what we call convergent evolution.
Convergent evolution.
Sure.
So different fungi coming up with similar solutions independently.
Precisely. Different unrelated fungal lineages developing very similar tactics to solve the exact same evolutionary problem. It's fascinating.
Okay, so what's the first category? You said one was particularly dramatic.
Yes, the predatory nematophagous fungi. So think about this underground battlefield. Nematodes are abundant, they're mobile. How does a stationary fungus catch these microscopic worms?
Good question.
Well, they produce these extensive networks of thread-like structures, the hyphae. Together, that forms the mycelium. That's basically the fungal body spreading through the soil. And it's along these hyphae that they develop specialized trapping devices.
Trapping devices. Okay, now this is where it gets really interesting.
It really is. The variety is quite spectacular. Each one is like a microengineering marvel. First, you've got sticky knobs. Imagine tiny, single-celled, round structures.
Like little lollipops.
Kind of like sticky lollipops, yeah. Covered in adhesive, spaced along the hyphae. A nematode brushes past, gets stuck.
And it's trapped.
Well, it gets stuck, and sometimes you can even pull the knob off its stalk. But here's the clever bit. The fungus can still penetrate and kill the host from that detached knob.
No way. So it doubles as a dispersal method, too.
Exactly. A way to spread itself while still getting its meal. Pretty smart.
Okay, what else is in their toolkit?
Then you have adhesive networks. Picture three-dimensional sticky nets. They're formed by hyphal tips looping back and fusing, creating this web that lifts slightly off the main fungus body.
So it's like an actual net?
Very much so. If you were looking under a powerful microscope, you'd see the whole surface just shimmering with this potent, invisible glue. A nematode thrusts its body into this web, and bam, it's quickly immobilized.
Okay, sticky knobs, sticky nets. What's next?
Next up are non-constricting rings. These are simple loops, usually 3 cells, formed by the fungus. The inner surface is sticky.
So another sticky trap.
Yeah, but passive. A nematode might push its way into the loop and just get tightly wedged, can't back out. Sometimes a struggling nematode can detach the loop, but just like the knobs, the fungus can still infect from the detached ring. No active squeezing here, though.
Oh, okay. So that implies there are active traps.
Oh, yes. And they are the most dramatic. The constricting rings. Again, these are 3-celled loops. Similar look to the non-constricting ones, but with a critical difference.
Which is?
Upon mechanical contact, just a nudge from the nematode on the inner surface, those 3 cells inflate incredibly rapidly
How rapid?
in about 0.1 seconds.
Seriously, 0.1 seconds.
Yeah, it's astonishing. This rapid inflation completely closes the hole in the ring, just like a noose tightening, severely constricting the nematode.
How does that even work?
It involves this super fast internal signaling, a biochemical cascade that triggers massive water uptake into the cells. The cell volume increases threefold almost instantly. It's this incredible hydraulic power on a microscopic scale, almost explosive.
That is absolutely incredible. Such precision and force in something so tiny. Now, you said most of these predators are Ascomycota, like Arthrobotrys.
That's right, Generellas like Arthrobotrys, Drechslerella.
Yeah.
But it's not exclusive. Some Basidiomycota get in on the action too. Things like Pleurotus, the oyster mushroom genus, and Hohenbuehelia.
Oyster mushrooms hunt worms.
They do, but with a different twist. They use these minute, lollipop-shaped cells, but instead of glue, they secrete drops of a neurotoxin.
A toxin.
Yeah, like linoleic acid. It paralyzes the nematode first, then the fungal hyphae move in to colonize it.
Wow. So why are they doing all this? Just for food?
Primarily, yes. They use the nematodes as a nitrogen supplement. Think of it like a protein boost, especially for those fungi that are also good saprotrophs, meaning they can break down dead organic matter too. It's an extra nutrient source when needed.
Okay, that covers the predators. What's the next category?
The next group are the endoparasitic nematophagous fungi. Now, unlike the predators, these are obligate parasites.
Meaning they have to live inside the host.
Exactly. They don't develop extensive mycelium out in the soil. They exist mainly as spores. These spores have 2 main strategies. Either they attach to the nematode's outer skin, its cuticle.
Like the sticky knobs, but just a spore.
Sort of. A good example is Drechslerella coniospora. It has these adhesive, knob-bearing spores, conidia, that often attach very specifically to the nematode's mouth end, like a tiny grapple hook finding just the right spot.
Very precise. And the other strategy?
Ingestion. The nematode accidentally swallows the spore. Harpisporium angululi is a classic example. It has these crescent-shaped conidia that, once swallowed, get lodged in the nematode's esophagus.
Ouch. And then what happens?
Once inside, whether attached or ingested, the spore germinates, grows mycelium throughout the nematode's body, consumes it from within, and eventually, often only reproductive hyphae emerge from the dead host to produce and disperse more spores. It's quite efficient.
Efficient but grim. Okay, and the third category.
Finally, we have the parasites of eggs and cysts. These fungi are generally considered opportunistic saprotrophs.
So they can live on dead stuff, but they'll take an opportunity.
Right. They're often found in soil or around plant roots, and they'll actively colonize nematode eggs, or those tough, hardened cysts we talked about earlier. This group is particularly important from an agricultural perspective.
Because they target the eggs and cysts of those damaging plant parasites.
Exactly, like the root knot and cyst nematodes. So they offer a potential natural defense against crop damage. Key examples here include fungi, like Pechonia clematisporia and Basidiomycetes lacinus.
It's incredible, the variety of strategies, but how do these interactions actually work? How does the fungus even know a nematode is nearby?
That's where the nematode-fungus interactions get really fascinating. It reveals some sophisticated communication happening at this micro level. Take trap stimulation, for instance. Many of those predatory fungi, they don't just form traps all the time in a lab dish.
They need a trigger.
Precisely. You add nematodes, or even just the water nematodes, have lived in their chemical exudates, and boom, within a day or two, traps start forming. Those exudates contain small molecules, like peptides, specific chemical signals.
The fungus is literally smelling its prey. It can sense these chemicals.
It seems very much like that. It tells us they're incredibly attuned to their chemical environment. They're not just waiting passively, they're responding.
Okay, so they sense the prey. Then what about actually sticking to it? You mentioned the glue.
Yes, the adhesion itself is remarkable. That glue on the traps and the spores, it's highly specialized. Research indicates it involves proteins called lectins.
Lectins, what do they do?
Lectins bind to specific carbohydrate molecules like sugars on the surface of the nematode. It's like a molecular lock-and-key mechanism.
So that explains the specificity you mentioned earlier, like Drechslerella sticking to the mouth end.
Exactly. That surface must have the right carbohydrate keyhole for the fungal lectin key. It ensures a strong, targeted grip.
And sometimes they use toxins too, not just glue or constriction.
Right. Beyond the physical trapping, some produce potent toxins. We mentioned Pleurotus using neurotoxins. Arthrobotrys oligospora, another predator, produces linoleic acid. It's highly toxic, causes hyperactivity in the nematode, then paralysis.
Paralysis first, then infection.
Makes the whole process easier for the fungus. And another clever trick, many nematophagous fungi also produce antibacterial antibiotics.
Why antibiotics?
It's likely a substrate defense strategy. Keep bacterial competitors away from their hard-won meal. Imagine you catch dinner, you want to make sure no one else steals it.
Makes perfect sense. Okay, so the nematode is caught, maybe paralyzed. How does the fungus actually get inside, the infection process?
In the predatory species, penetration often happens via a specialized structure called an opressorium, or just a hyphal tip. And this then immediately swells up inside the nematode's body to form a globose structure called an infection bulb.
An infection bulb, like a beachhead.
Exactly like a beachhead.
Huh.
From this bulb, feeding hyphae, the assimilative hyphae, spread throughout the nematode, absorbing nutrients.
And the endoparasites, the ones that live inside.
They typically don't form that initial bulb. The nematode might actually stay alive longer while the fungal hyphae proliferates inside, slowly consuming it from within. A slightly different, perhaps more insidious approach.
All this incredible biology, does it have practical uses? You mentioned agriculture.
Absolutely. All this science points to a huge potential in biological control. These fungi hold significant promise for controlling harmful nematodes, both in agriculture and potentially in animal health too.
Like using fungi instead of chemical pesticides.
That's the hope. For example, there's one species, Arthrobotrys eudermata. It's unusual because it forms these really tough, thick-walled resting spores called chlamydospores. These spores are resilient enough to actually survive passage through an herbivore's digestive tract.
So you could potentially feed them to livestock.
Exactly. To control parasitic nematodes living in the gut or passed out in manure. Imagine a future where we harness these microscopic hunters to protect crops and animals naturally, reducing our reliance on chemical interventions.
That sounds amazing. Are there challenges?
Oh, sure. Practical challenges remain. Things like difficulties in mass-producing the fungi effectively or ensuring they survive and compete when introduced into new soil environments. It's not always straightforward. But connecting that with the bigger picture, the potential and the sheer ingenuity are undeniable.
Okay, from that hidden battlefield beneath our feet, let's completely change scene. Let's plunge into a different kind of challenging environment. Fast-flowing water. You mentioned fungi in streams.
Yes, let's talk about the masters of the flowing waters. The aquatic Hyphomycetes, or sometimes called Ingoldian fungi.
Ingoldian fungi.
Named after the pioneer C.T. Ingold, who really brought them to light. If you were to take some foam from a fast, babbling brook, especially in autumn, and look under a microscope,
What would I see?
you'd find this bizarre and beautiful world of fungal spores, unlike anything you typically see on land.
Bizarre and beautiful how? What's special about them?
Well, it's primarily their extraordinary spore shapes. Over 300 species have been described, but two common unique shapes really stand out. First, there's the tetraradiate conidium, think tetra for four, radiate for arms spreading out, like a tiny branch star or grappling hook with four arms.
Okay, a four-armed star. What's the other shape?
The other common one is the sigmoid conidium, sigmoid meaning S-shaped or sometimes described as a steep helix, like a curved sickle or a very bent letter S.
Star shapes and S shapes. Why these specific forms?
That's the fascinating part. These precise shapes are prime examples, again, of convergent evolution. Unrelated fungi, living in this challenging environment of flowing water, have independently evolved these very same complex shapes.
Because those shapes help them survive in the current.
Exactly. They all face the same problem. How do you attach yourself to something, like a leaf or twig, when the water is constantly trying to wash you away? These shapes are the solution.
Okay, let's break that down. The significance of the spore shape. How does the tetraradiate, the star shape, work?
Imagine that microscopic throwing star, or maybe an anchor. When it tumbles through the water and hits a surface, like a submerged leaf, its shape almost guarantees a three-point landing. Two or three arms make contact almost simultaneously.
Which makes it stable?
Very stable against the current. And then there's mucilage, a sticky substance at the arm tips. It acts like a quick-set glue, forming specialized attachment pads, brissoria, that anchor it firmly, resisting detachment. You can picture species like Articulospora tetracladia with these elegant jointed arms perfectly designed for this.
Okay, stable three-point landing for the stars. What about the sigmoid, the S-shaped spores? They sound less stable.
They generally trapped less efficiently, that's true,
Yeah.
but they have their own strategy. As these curved spores approach a surface in the current, they tend to tumble end over end. They might initially make contact with just one tip. Then the force of the current causes them to pivot, swinging around until they lie parallel to the surface in the flow.
Ah, minimizing resistance.
Exactly. Minimizing the shear forces trying to rip them off, they end up achieving a stable two-point contact along their length. So, both shapes, though different, are elegant solutions, ensuring the spores get efficiently trapped onto submerged surfaces, preventing them from being swept away downstream.
It really makes you wonder, how did these specific complex shapes evolve over and over again?
It really speaks to the power of natural selection in that environment. The selective pressure to anchor effectively must be immense, pushing different lineages towards these optimal forms.
So they're good at sticking around, but what are they actually doing there? What's their ecological role?
This is where they become absolutely crucial, really vital to the whole stream ecosystem.
How so?
Think about streams flowing through forests. Where does most of their energy, their carbon, come from?
Falling leaves, I guess.
Primarily, yes. Tons of autumn leaves fall into streams. But here's the problem. Those leaves initially are tough, low in nitrogen, and pretty unpalatable to the aquatic invertebrates, the little critters at the bottom of the food chain.
So the insects can't eat them directly?
Not easily, and they don't get much nutrition if they do. This is where the Ingolian fungi come in. They are like the stream's primary decomposers and conditioners. They colonize these fallen leaves. They have powerful enzymes, cellulose, pectinases, that start breaking down the tough leaf tissues, softening them up.
Pre-digesting the leaves.
Exactly. They're pre-digesting them. And crucially, as the fungi grow, they enrich the leaves with their own microbial protein, boosting the nitrogen content. They turn low-quality roughage into a much more nutritious food source.
So they make the leaves edible and nutritious for other things.
Precisely. Aquatic invertebrates, like mayfly nymphs or caddisfly larvae, show a strong preference for feeding on leaves that have been colonized by these fungi. And studies have shown it clearly. Animals fed a diet of fungus-colonized leaves show much greater growth, they gain more weight, and they're more fecund. They reproduce more successfully than animals fed only plain leaves.
Wow, so these fungi are basically feeding the entire stream food web.
In a very real sense, yes. They are essential intermediaries. They unlock the energy and nutrients in those dead leaves, making it available to the invertebrates, which are then eaten by fish, and so on. They are absolutely fundamental to maintaining the productivity and health of these stream ecosystems. Unsung heroes, really.
That's a huge role. And besides the spore shapes, do they have other special adaptations for living in water?
They do. As mentioned, they have that broad range of enzymes for degrading tough plant material efficiently. Many can also grow at surprisingly low temperatures, even near freezing, which is vital for cold streams, especially in winter or mountains. And intriguingly, some can survive for quite a while on dried leaves.
Dried leaves. How does that help in a stream?
It suggests they might be dispersed by wind between different water bodies. A dried-up leaf blows away, carrying the fungus, and if it lands in a new stream, the fungus can rehydrate and start colonizing again. Adds another layer to their dispersal strategy.
Amazing adaptability. Okay, let's shift gears one more time, from fast streams to
What?
slow water.
Exactly. Let's move to another, even more specialized, aquatic niche, stagnant ponds, slow-moving ditches, places with very little water flow. Here, we find a different group, the aeroaquatic fungi.
Aeroaquatic, air and water.
Precisely. They're known for their unique bubble trap propagules.
Bubble trap propagules, what does that mean?
Well, their key characteristic is fascinating. These fungi do their vegetative growth. They build their mycelial body while completely submerged, often in muddy sediments with very low oxygen levels. But they only sporulate, they only produce their reproductive structures after they get exposed to air.
How would that happen? Like the pond drying out at the edges.
Exactly. At a miced interface between water and air. And critically, the spores they produce, their conidia, are specifically designed to trap air.
Why trap air?
So they can float. If that substrate, that leaf or twig they're on, gets resubmerged when the water level rises, the air bubble keeps the spore buoyant, floating on the surface. It's their dispersal strategy for still or slow-moving water.
Okay, that's clever. What do these air-trapping propagules look like? Are they all the same?
Oh no, there's an amazing variety, showcasing convergent evolution yet again. Different solutions to the same problem. Some form these elegant cylindrical or barrel-shaped spirals. The genus Helicune is a great example. Imagine a tiny, perfectly coiled spring made of fungal cells trapping an air bubble in the middle.
Like a little life raft.
Pretty much.
Right.
Others form structures that look like tiny balloons, sometimes single-lobed, sometimes bilobed, Hohenbuehelia pulmonaria does this, using aggregates of dark, thick-walled cells to enclose an air cavity.
Balloon fungi? What else?
And then, perhaps the most intricate are species like Clephosphaerina zalewski. They create these amazing hollow spherical structures. The walls are like an open lattice, almost resembling miniature practice golf balls or geodesic domes.
How do they build that?
By their hyphene repeatedly branching, curving back, and fusing together in a very precise pattern to create this hollow, air-filled sphere.
It's just incredible. Different groups, different lineages, all figuring out ways to trap air for flotation.
And their ecophysiology, how they function in that environment, is equally remarkable. As we said, these bubble trap propagules are hydrophobic. They repel water, which helps them float effectively. They colonize autumn leaves rapidly when they fall into ponds. And then they continue to grow underwater, often in anaerobic or oxygen-depleted conditions.
They can survive without oxygen.
They show incredible survival capabilities in anaerobic conditions, yes. Even when there's toxic hydrogen sulfide bubbling up from the decaying mud at the bottom of the pond, they can survive like that for months sometimes.
It's extreme survival.
It absolutely is.
Yeah.
It's a critical adaptation for life on stagnant pond bottoms. Then, sporulation is often triggered when conditions change, when they get exposed to aerobic conditions, maybe some light, after being submerged for a long time.
Which raises the question, right? What are the molecular mechanisms? How do they switch their metabolism back and forth between oxygen-rich and oxygen-poor conditions so effectively?
That's still an active area of research. Understanding how they thrive and manage their energy in such extreme, fluctuating environments is a key. Well, I think this deep dive into these anamorphic fungi really reveals a microscopic world of just incredible specialization and adaptation. It shows how diverse and resilient life can be, even in the most challenging spots.
Yeah, definitely.
We saw three central themes pop up again and again. First, that idea of convergent evolution, how completely different fungal lineages independently came up with similar, really sophisticated solutions. Think of the nematode traps or those specific spore shapes in the aquatic fungi. Same problem, similar answers evolved separately.
That really highlights the power of the environment shaping life.
Absolutely.
Oh,
Second, their immense ecological significance. We often overlook fungi, but these groups are critical. They regulate nematode populations, maybe helping us with biological control one day. And in streams, they're essential nutrient recyclers, turning dead leaves into food that fuels the entire aquatic food web. They're not just there, they're doing vital jobs.
yeah, the stream example really drove that home. They're foundational.
Foundational, exactly. And third, just their extreme adaptations, their physiological ability to not just survive, but thrive in wildly fluctuating conditions,
Sure.
whether it's the physical force of a fast stream or the chemical challenges, the lack of oxygen in a stagnant pond mud. Their survival toolkit is just extraordinary.
It really is. The sheer ingenuity of these organisms, it just reminds us that even in the most specific, difficult niches, life finds a way. It adapts, it flourishes, and it plays a vital, often completely unseen role in the bigger ecological picture. It definitely invites you, our listener, to consider the hidden wonders of the microbial world that are literally all around us, under our feet, in the water, constantly working, adapting, innovating in ways we're only just beginning to truly appreciate. It certainly makes me think twice about what's going on in a handful of soil or a drop of pond water.
Well said. Thank you for joining us on this journey of discovery today.
Yes, thank you. Until next time, keep exploring, keep questioning, and keep digging deeper. From all of us at The Deep Dive, thank you for listening.